All right, Biology 11 students, our next topic is the origin of life. How did life start on this planet of ours? A big topic. We are going to break it down into a series of steps or events that are hopefully, in each little step or event, presented in a way that makes it understandable so you can understand how we got to the, how we get to the next step or event. All right? So, of course, if we're talking about the origin of life, we have to talk about the planet before life was even on it. And that planet was called the Primordial Earth. Right? The Earth is 4.6 billion years old, scientists believe. That's billion with a B. So the Earth formed approximately 4.6 billion years ago. The newly formed Primordial Earth, very, very harsh place. Very mean to life. Right? An inhospitable planet at this point. Tons of volcanic activity, superstorms, tons of rain once it cooled down. Just if we could take a time machine and go back to the primordial Earth, as soon as we left the safety of our time machine, we would probably die. Right? So for a long time, there was no life on this planet. When it was newly formed, there was very little of anything that would support life. It was way too hot, way too violent a place. So it was so hot because asteroid impacts created a ton of heat, internal compression of all this, all the gravity taking, effect on all the molecules that were now gathering to form the planet. And then, of course, radioactivity. We didn't have that ozone layer yet. So the radioactivity and radiation that came from the sun would also, add a lot of energy to the planet. Just to cool down, this newly formed Earth took 500 million to 800 million years to do this. Just to cool down, and that was to get it to a temperature on the surface of the planet that was below 100 degrees. Not the comfortable, 20 degrees or 30 degrees we're used to, just to get it underneath of 100 degrees. It took about 500 to 800 million years, scientists believe. So a very long time. What that cooling would do is, once you get underneath 100 degrees Celsius, water no longer boils and turns into steam or vapor. It condenses. And so you would have rain, forming and falling and gathering in the low-lying areas and forming lakes and oceans. And this is important because all biologists agree that this is where the life would have formed from first, right? Life arose, started in the water and arose and eventually moved its way onto the land. But we needed that water first. The cooling of the planet provided that liquid water, not a water vapor. So the young Earth, the primordial Earth, ton of volcanic activity, as I mentioned before, and this would impact the atmosphere and the temperature. It was still a very hot planet compared to what we have now. The atmosphere of the primordial Earth would have had a ton of nitrogen gas, so that's N2. It would have had a lot of carbon dioxide. Volcanoes spew out a lot of carbon dioxide. Right? When there's a volcanic eruption, they tell people get away because obviously you don't want to get hit with ash or lava that's really, really hot. But what happens when a volcano erupts, an invisible cloud of carbon dioxide comes out of the top of it and runs down the side of the volcano. And they have found creatures that have asphyxiated close to volcanoes that did not die because they were burned or a piece of debris hit them. They basically choked to death because of this invisible cloud of CO2. So with all the volcanic activity, there would have been a lot of CO2, as well as carbon monoxide, and of course water was there, we know that. And another gas, methane. Methane is CH4. That's probably the one you may not have known the formula for. Ironically, in this primordial Earth, there was very little atmospheric oxygen. So again, most of the stuff on the planet today are aerobic, right? They need oxygen to make their energy, their ATP. The primordial Earth was having none of that. Lots of other things, but very little oxygen. So we couldn't go back there. We would have choked, we would have asphyxiated and died pretty quickly. Lots of things would have. But if you look at these materials, you have the building blocks for life, right? In terms of organic chemistry, the big four, right? In terms of biomolecules, the big four are carbon, which we can see came in a variety of compounds. Hydrogen, we've got a couple of sources here. Oxygen, now not atmospheric oxygen, just the element oxygen, which we have here and here and here, of course. And nitrogen, which would be in the nitrogen gas. Our atmosphere is still 78% nitrogen gas, so there's still a ton of that. And if you throw a little phosphorus and sulfur, and there were sulfur beds back in the primordial earth as well. They were like hot springs, only boiling mud and sulfur. There's still a few on the planet today. If you give me these four, I could probably make 90% of what's inside of your body. You add these two elements as well, which were on the primordial earth, and you could probably make 97, 98% of what is inside of you, what makes up your body, right? So all of the biomolecules, you give me these 6 elements, I can make up an awful lot of stuff that helps support a living thing. So, and that just kind of, this point goes along with that. Anyway, so here are a couple of pictures of the primordial earth. So I got these off of a video. There's a YouTube video where scientists have put together like this little 35 second piece on YouTube showing the primordial earth. And it actually looks really, really cool. It looks like something out of an Avengers movie. But again, the atmosphere and the temperature and the landscape, just not hospitable for life at all. Here's another one here. It's some volcanic activity. You can see the lava fields running. Just not a really nice place for any living thing at this point in time. So the planet was devoid of life, right? Very inhospitable, no life. And today there's millions of life forms. So at some point we had to have life arise on a lifeless planet. So how did that happen? You know, where does life begin when there is no life previously there? What we look at is a theory called primary abiogenesis. Now we know primary means the start of something, the initial, right? The beginning. And then there's abiogenesis. Now A means not or none, right? Bio is life and genesis is the creation of. So basically what this is saying, it's the start of life from a place where there's no life there to create it from. Right? So that's what that kind of means there. So what we're going to be doing is basically taking all this inorganic stuff. So all of these are inorganic. And we're going to try to make organic stuff from them. Organic molecules are the high energy ones that fuel your body and build your body and repair it and stuff like that. So we have to get from this to that. Now organic structures are larger and they carry more energy. So they have a higher energy value. These ones are smaller. If you look at a lot of these, 5 atoms, 2 atoms, 2, 3, and 3 atoms. So not very big. Right? An example of an organic structure is like glucose, C6H12O6. That has 24 atoms. That's significantly larger than any of these. But to go from a small molecule to a large one, I need energy, right? To build the bonds and to make the structure. Think about it. I could give you a bunch of boards and bricks and glass and that, and to build a house, a much larger structure out of those building blocks, you have to put in a lot of sweat equity. You've got to put in a lot of energy to build the larger house structure. It's the same with chemistry. To build a larger molecule, you've got to input a lot of energy. So you need the building blocks. We got those because we said the big 4 were C, H, O, and N. And we need a source of energy, which I'm going to kind of just draw a little lightning bolt here. If we have those two things, we can go from inorganic to organic. So The energy was, well, we talked about the megastorms, the lightning, the radioactivity, the heat of the volcanoes. That provided our energy, and of course the inorganic stuff provided that. So could it be done? Well, we wondered about this, and then there were two scientists, Stanley Miller and Howard Urey, 1953, the University of Chicago. They ran a very simple experiment that showed, yes, primary abiogenesis could have happened. We could go from an inorganic planet to one that had organic molecules capable of building life on it. All right? So here's a picture of their experiment. Now, if you go to YouTube, you'll be able to find a video of this thing, kind of an animation of that thing. But basically, here's the rundown of how it was set up. They had water here with heat underneath it. You can see a Bunsen burner. And that would create water vapor, which would circulate around through this apparatus. So the water vapor would go up here and then over, and it would gather in this bulb here. Now, in that bulb, they had methane, so CH4. They had ammonia, another one of the gases. I forgot to write ammonia in here, but ammonia would have been present in the primordial earth, another small inorganic molecule. So ammonia, and they had hydrogen gas, another gas that would have been present. It's another inorganic molecule, so H2. And of course, they had the water circulating. So if you look at it, between these four underlying substances, I've got my C, I've got H in all of them, I've got O in my water, and I've got N from my ammonia. So I've got all four of the big building blocks circulating around this device. Now, to simulate the lightning and the radiation and the volcanic heat, what they did was they had a couple of electrodes that would flash inside of this bulb that contained the gases. So these molecules were all in here, and there is basically, with the two electrodes, miniature lightning, right? So this mimicked the energy needed. So we had the four elements and the lightning inside of it. Whatever was formed in here, you know, these small substances might fuse together, they would follow down here, the water would go through a condenser, and all that happens here is that cold water goes in and takes the water vapor and turns it into liquid water, because the cold water drops the temperature of the material that goes down through the condenser. So we had liquid water here, and then they had a collecting flask here. And they ran this thing for about a week. And what did they find? So there's the setup. There's a brief outline like I just described to you. So what were the results? Well, they ran it for about a week, and they examined the water that they collected in this little flask. And inside of it, they found two types of amino acids, glycine and alanine. So 2 of the 20 amino acids were there. Amino acids are the building blocks of protein, if you remember. They found urea, another organic compound related to protein. They found aldehydes and carboxylic acids. These are all organic molecules that are larger than these ones, right? So what they were able to basically do was start off with an inorganic lifeless experiment and not create life, but at least create some of the organic building blocks that would allow for life to at least get a foothold, if we can get the right combination of these building blocks together. All right? So since Miller and Urey did their experiment in the 50s, of course, with our technology and our scientific understanding ever expanding, we've been able to run this experiment with even greater technology, and they've yielded, now when we do it, we get simple sugars like glucose, all 20 amino acids. There's 20 different amino acids that are used to build all of our proteins, which run and regulate and build our body. They've created all 20 with this similar apparatus to this, with the similar theory behind it. Many different types of vitamins, and all 4 of the nitrogenous bases of DNA. That's the A, T, C, and G that we talked about in class. Adenine, thymine, cytosine, and guanine. They've created all of this with setups very similar to Miller and Urey's from the 50s. So definitely you can go from a lifeless planet to one that at least can create the molecules for life. Now, the first cells. The first cells were prokaryotic cells. Obviously, bacteria are the simplest life form. So it kind of makes sense that we're going to build bacteria out of this inorganic soup before we go and build things like elephants and dinosaurs and people. All right? The first cells were prokaryotic cells, and they form structures called stromatolites. And when scientists go and look at these fossils, these remains, these stromatolites, they carbon date them, and they found out that they were 3.5 billion years old. So if you look at it, the planet's 4.6 billion years old, and life is 3.5 billion. Not us, not the dinosaurs, and not the your dog and that, but the original life, those early prokaryotic cells. So it took almost 1 billion years just to get to the point from the Earth's formation to something living that we could call life. The stromatolites are bacteria, and they're very similar to cyanobacteria. Cyanobacteria is a photosynthetic bacteria. We still have them on the planet today, and they run photosynthesis. And these ancient photosynthetic bacteria that form the stromatolites would do two very, very important jobs, right? The first thing they would do is they would convert small inorganic molecules into larger energy-rich ones. Think about a plant, right? These things were photosynthetic. So what they do is they take in CO2 and water, and what does a plant do with that? It creates oxygen and glucose, a simple sugar, right? We know glucose is C6H12O6. And the planet takes in these molecules, and it uses the energy from the sun to do that. Now think about it. These are the only living things right now, these bacteria that are running photosynthesis. They're taking small 3 atoms and 3 atoms, building blocks, and creating a much larger one. And to do that, they have to have energy. This is a high energy compound, and the energy came from the sun. The sun acted like a welder's torch and fused these together, and now that energy is locked into the structure. So we've got organic molecules being built up. What they also do, remember, these are the only living things right now. They're running photosynthesis for years and years and years. What they're doing, the second thing is, they're removing this from the atmosphere. So this is going down, and this is going up, right? So CO2 levels are dropping because the CO2 is getting converted into glucose, and oxygen levels in the atmosphere are increasing. And if you allow these things to do it over millions and millions and millions of years, what you will see is an atmosphere that has a ton of carbon dioxide. Those levels will drop, and currently today it's less than 1%, our atmosphere. And what we see is the oxygen levels, which we said earlier, there was hardly any free atmospheric oxygen. It increases dramatically. And currently today, our oxygen levels are 20 to 21%, right? Probably closer to 20 with the amount of combustion that we do. So if you look at it, to make ATP, which is what we need to do, we need 2 things. We need oxygen, and we need a high energy fuel source. We run cell respiration with this, with these 2 materials, and we make ATP, which is cellular energy. So this is huge, these first prokaryotic, early photosynthetic bacteria doing this. They open the door for much larger life forms to eventually take hold, much more complex ones. So how do we go from a prokaryotic cell, a simple single-celled bacterial cell, to eukaryotes, like protists, fungi, plants, you, me, dogs, cats, and all of that, right? Well, we have our early cell, right? It's a prokaryotic cell. And what scientists believe, and I'm just going to erase this, what scientists believe is that in that early cell, we basically had, and there's a much better picture coming up than what I'm about to draw. So here's our early prokaryotic cell. And what they believe is the cell membrane is very soft and pliable. So if you push in on the cell membrane, you can actually create indentations. All right, so if I pushed in on the side of this thing, it would indent. So what they think may have happened was the cell would push in like this, and it would indent here like that, it would indent here like that, and indent here like that, like that. Now our DNA is in the middle here, right? Because all cells have DNA. An early prokaryotic cell would have the circular ring of DNA, right? I'll try to draw a circle neatly in there. And what they believed happened, right? If you remember, a prokaryote, there's no nucleus. The early eukaryote would have to have a nucleus. Eukaryotic cells have a nucleus. And they believe these phospholipids, what the cell membrane is made out of, would have simply fused together just like this. And then these would have sealed around like that. And our phospholipid bilayer, it's very fluid and it can do this, right? It's like a soap bubble. The surface is capable of moving around. So if you look at it, that would have been the formation of an early nucleus, right? And then smaller indentations that would have occurred would have formed things like, and then they seal off, the Golgi apparatus or the ER, vacuoles, all different things would have formed. And if we look at it, the membrane of the Golgi apparatus, the ER, the vacuole, it's all made of phospholipids. 2 layers of lipid molecules, which we see here. So here's a picture of a cell. We can see the cell membrane and the phospholipid bilayer. Phospholipids are the building blocks of the cell membrane, and it's a bilayer, which means 2 layers of them. We'll get into the chemistry of why there's 2 layers in grade 12. But here's a picture here. There's 1 layer of phospholipids and another one here, and they make up the cell membrane. When we look at the Golgi, the vacuoles, the ER, and a variety of other organelles, they have a phospholipid bilayer that matches perfectly what's in the cell membrane. So they believe that this pushing in and inward infolding of the cell membrane could lead to the formation of these other organelles like the nucleus, the Golgi, the ER, and so on. So here's a really good picture. You can see here, here's our early prokaryotic cell. And here's the, much better picture than this of the formation of the nucleus here. You can see how there's little indentations of the membrane, the external cellular membrane, that would surround the DNA, and then it would pinch off, and now you've got an early nucleus. More infolding would occur and form things like your ER and your Golgi and that. And then the second half of this diagram, these last 4 cells here, we're just about to get into. This is endosymbiosis, one of our next topics. Because we know that prokaryotic cells had very few organelles. Eukaryotic have a whole bunch. So we explained how some of those organelles, like the nucleus, ER, and Golgi, came about, vacuoles. But they also have a couple of key players, mitochondria and chloroplasts. These are the big energy organelles. Chloroplast runs photosynthesis, that creates glucose, big energy, and then the mitochondria takes the glucose and makes ATP, big energy. So this is an important step right here. It's called endosymbiosis, and it's the next topic of our slide. So, if we look at endosymbiosis, the term endosymbiosis. All right? The cis on the end just means a process. Whenever you see that, cis means process. In grade 9, I believe, you do symbiotic relationship. So here you're living in association with something else. So I'll just say a roommate. And endo means enter or to come in. All right? So basically, this is the process of something entering the cell and living in a close association with it. All right? Now, if you remember our symbiotic relationships, there were 3 of them. We're going to focus on one of them. The symbiotic relationship that we're going to look at is mutualism. Mutualism is where the 2 things live together, and organism A receives a benefit and organism B gets a benefit as well. The other 2, if you're wondering, where parasitism is one gains, one loses, and commensalism, where one gains and the other's unaffected. But this one was a mutualistic one, all right? So researchers believe that the process of endosymbiosis accounts for the presence of mitochondria and chloroplasts, all right? So I'll draw the mitochondria in blue here. Our mitochondria looks like this, and then there's an inner membrane that looks like this. It's highly folded, right? And this is what makes ATP, right? If I draw the chloroplast, I'll draw the chloroplast in green because it's a plant organelle. The chloroplast looks kind of like this. There's two membranes again, and then it has these little photosynthetic reaction centers called thylakoids. We don't have to know thylakoids. Grade 12 you will. And they're connected by little lamellae bridges, but stuff that you'll have to know for grade 12. And of course we know this one runs photosynthesis. This is the chloroplast, and this one makes glucose. That's the big energy payoff from that. All right, and it also kicks out some oxygen, not too bad. All right. Those darn plants, we should cut them all down. What do they do for us anyway? Glucose, oxygen. Anyway, so this would only add to the abilities. Once these two things entered the cell and got into a symbiotic mutualistic relationship, the cell would be able to harness a ton of energy because it could absorb, if it got a chloroplast inside of it, it could absorb sunlight now and make glucose. And if you had mitochondria, you could take the glucose and make ATP. Energy galore. And that energy will allow us to go from single cell to multi-cell to cellular specialization, right? And eventually lead to what we are. Something that's made of 100 trillion cells that are all very, very, specialized. Couldn't do that without this step happening. So what scientists believe are these things are little prokaryotic entities that would have been engulfed or that would have invaded the cell, right? So you have basically what it's saying is I'll try to draw this nice and neat. We have our early eukaryotic cell. And our little mitochondria, which are very, very small, there's one there, or our little chloroplasts would have somehow gotten inside of these early eukaryotic cells. So somehow they got drawn in, right? Now some cells would have taken both of them in. Other cells, it was found out, only took in the mitochondria, right? Now, inside of these cells, we still had the nucleus, we still had this ER, we still had the Golgi and vacuoles, right? All that sort of stuff was all there, right? A variety of other organelles. And they would have went in. Basically, the cell reaches out and engulfs these things, thinking they're food, and then they realize, oh man, this is a win-win situation. I'm going to keep this thing. I'm not going to destroy it, right? So these things, when they come in, they would have been engulfed by a vesicle. A vesicle is made of the host cell membrane. So basically, if I draw that very quickly, I'll try to draw it. There's this. That's our cell. There's our mitochondria. The cell would reach out and engulf this thing like that with extensions of its cytoplasm. And then these two arms would eventually, these two arms right here, these pseudopods would connect together and fuse, and now we have this part of the membrane right here. This part of the membrane, the orange material here, is a vesicle. And so it already would have had one membrane here, and now it has a second membrane, the orange one, which would be here. Two membranes. The same thing would occur with the chloroplast. Here's its original membrane, and this is the new membrane from it being engulfed by that cell, right? This process is called phagocytosis. And that's what they believed would have happened, is these little bacteria, prokaryotes, got engulfed by the early eukaryotic cells. And then they found out just how good they are, right? So they would have engulfed it, probably thought, I'm going to break it down, use it for energy, and then realize, whoa, wait a second, this one's making a ton of energy, and this one's processing it and making even more. I'm not going to destroy these things. I'm going to keep them. It's win-win, right? So this is a mutualistic relationship, which we talked about already. And in their new home, now they're, these two, what do they gain? We know the cell gains energy, but these two now gain access to nutrients and protection that they never had before. So it's win-win for both, right? Now, the heterotrophic lineage, things that are heterotrophs like us and fungi and some of the protists, they would have only taken in the mitochondria. And the autotrophs, like your plants and some protists are autotrophic, would have taken both the chloroplast and the mitochondria. They would have taken both in. For some reason, a lot of students think animals have mitochondria, plants have chloroplast, and that's it. Plants have both mitochondria and chloroplast. So plants take in both. We only took in the one. So here's a picture of it here online. You can just see where you have your mitochondria and your chloroplast here, your early eukaryotic cell, the organelles are forming, there's our early nucleus. And then this would be our heterotrophic lineage here. They only took in the mitochondria. And this would be our plant lineage here. They took in both the mitochondria and the chloroplast. Right? And that's what this is basically saying, is that both of, the heterotrophs would have taken in just the mitochondria. The autotrophs would have taken in both the chloroplast and the mitochondria. Now, is there any proof of this even happening? Right? Well, an easy way to remember the proof is Mad Doctor, M-A-D-D-R. And I was able to find a picture of, is he a professor or doctor? I guess he's doctor, right? Mad Doctor. Doofenshmirtz from Phineas and Ferb. One of the few cartoons that I actually have time for. Anyway, the M. So these 5 letters each stand for a piece of evidence that this process actually took place. M is for membranes. Most organelles have one membrane. So there's one membrane. Golgi, ER, vacuole, all that. These things have 2. And that second membrane would have come in through phagocytosis, which is a form of endocytosis, entering into the cell. That second membrane is this orange circle around, well, I drew mitochondria here, so the orange circle around it. Again, one membrane here and a second one here. That second membrane suggests that this thing was adopted by the cell through this process. Antibiotics. Antibiotics are things that kill bacteria, right? Like penicillin, vancomycin, ampicillin, cephalosporin. These all attack bacteria. Well, most of the stuff in our cells just kind of looks at it and goes, oh, awesome, go kill the bacteria. These things are susceptible and can be hurt by antibiotics. So that indicates that they could be bacterial in nature. This is a big one here. So DNA. These things actually have their own DNA. And DNA we know is the blueprint of life, which means these must be living on their own. So if you look at it, even though they're in our cells, we have mitochondrial DNA, right? Each of your mitochondria has its own DNA. And we know that DNA is a genetic code for living things. It's the instructions for a living thing. So this is a living thing, right? And if we look at it, the DNA inside of here, it doesn't have a nucleus either. It's just a small circular piece of DNA, which is the same as bacterial DNA. Ours comes in strands called Chromatin, which coils and condenses into 46 pieces called chromosomes, this has one circular piece of DNA. The other one, division. When these things divide, it's by binary fission, which is the same process that bacteria use. They follow the same steps. And the last thing, the R in Mad Doctor, they have ribosomes. And the ribosomes are not like eukaryotic ribosomes. The ribosomes inside of these things are not like the ones we have in the cytoplasm floating around in our cells. The ribosomes inside of these two things are the same, exactly the same as those that are floating around inside of other bacteria, free-living bacteria. So there's quite a significant amount of proof. Here's our tree of life here. We're almost done. So here is the universal common ancestor. And we can see the archaebacteria here and the eubacteria here. And then we have the eukaryotes stemming from here. So the question always is, who are we most closely related to, the archaebacteria or the eubacteria, the new school ones or the old school ones? When we look at our cells and we look at their characteristics and we compare them to the archaebacteria and the eubacteria, we are actually more closely related to the archaebacteria, which is an odd thing, right? So we have more relation, more traits in common and more stronger relation to archaebacteria than we do to eubacteria, even though we are called eukaryotic. And I just thought I'd point that out so you can see a diagram here. And of course, when we did diversity, you'll recognize a lot of these terms now, which is great. We're expanding our knowledge. And that's it. Found this picture of Charles Darwin, and I thought it was really poignant. I really, really like this picture. Him thinking there, and looks like he's going shh, right? So anyway, that's the end of it. I hope it's nice and clear, making a little bit of sense to you. If there are any questions, comments, or concerns, please place them in the comments section of this YouTube video, or if you're one of my students, reach out to me through Edsby. And I'll talk to you soon. Bye-bye.